Good morning. This is Joseph from HKU Ophthalmology. Today, I will share with you briefly about our ongoing work on the use of microfluidic technologies towards the detection of biomarkers of ocular diseases within eye tears. Recent research has focused to reveal the possibility of using eye tear as the next routine body fluid test to diagnose ocular diseases, as tear has less complex composition than blood and can be collected non-invasively. Therefore, recently, a lot of proteomic analysis studies have tried to identify many potential disease biomarkers using mass spectrometry. However, to validate these potential biomarkers to be clinically relevant, large-scale clinical studies are required. Relying on mass spectrometry is not so cost-effective, and will also require skilled personnel to run the analysis. Therefore, an alternative analytical method, digital ELISA, may be a possible solution. I think most of us are familiar with ELISA, which detects the target biomarkers by capturing them using respective capture antibody. After conjugating with a fluorescent tag, the complex will express fluorescent signals and we will get an analog-based result as the concentration of the target biomarkers. However, the limit of detection is usually to the level of picogram per mil, which may not be enough to detect the potential biomarkers in tear samples. Therefore, here where the digital ELISA comes. The capture site of digital ELISA is on microspheres that are covered with numerous capture antibodies of the target biomarker. If the number of microspheres and the concentration of target biomarkers fulfills the Poisson distribution, each particle will either bind with 0 or 1 target biomarker, and that's why it is called the digital ELISA. The theoretical limit of detection can be 1,000 times lower than traditional ELISA. By counting the number of beads with fluorescent signals, we can then back calculate the concentration of the target biomarkers. However, to fully transform the use of digital ELISA for tear analysis, we need to tackle the following issues, including the small volume sample of tear, the readiness of the instrumentation to general labs, and the tedious and time-consuming procedures for post-analysis. Therefore, we are now trying to develop a microfluidic device to perform digital ELISA for tear analysis that can quantitatively detect the target biomarkers in an easy, rapid, and economical manner. Our current two-layer microfluidic device have two major features: the bio-inspired self-driven capillary pumps, which facilitate the quick sample loading, and a high-density microgrid array chamber, which improves the efficiency in capturing microbeads in a denser manner for image analysis. With these features, we can then easily load the prepared samples using a pipette, and the sample will then be automatically infused into the device. To achieve an effective and automatic sample infusion, we incorporate the microstructures that can be found on the surface of a pitcher plant. Such structures allow a very effective automatic directional flow based on very strong capillary actions. From the videos, you can see the capillary effect to drive the samples with beads to flow along the channels. At the center of our device, there is a high-density array of microcavities, which is used to capture the floating beads. Such oblique microcavities are effective to capture and hold the floating beads, as shown in the videos. The capturing process takes only a few minutes, and we can then capture thousands of hundred beads within the cavity for our imaging analysis. Currently, we are now validating our device using commercially available digital ELISA assay kit. In here, I would like to thank my student Ziting, who is working on the project, and my collaborator, Professor Wu, who provides us the technical support on the device fabrication. Thank you very much for your attention.